Start of the talk is 10:40, so all those technical issues should not affect the schedule, I guess. Thank you so much for being here, and I would like to thank the organizers for giving me this opportunity to give this talk to introduce the research that we do in my lab. Let me see. All right, just as a matter of a short introduction, my lab works on a particular slice of protein bioinformatics related to characterization and prediction of protein structure or lack of structure and function at a residue level. So we'll be characterizing functional and structural properties of proteins for individual amino acids in a sequence. So it's in a way somewhere in between which you can collect from PDB, which is atomic level information, and something that you can largely collect from resources like UniProt, which is largely protein level characterization of protein structure and function. We do it kind of in between those two domains. And the focus of my talk will be on a slice of this research that we do related to the characterization and prediction of intrinsic disorder in protein sequences and the functions of intrinsic disorder. Oops. Working with disorder at the protein sequence level. All right. So what is disorder? I guess I will discuss it at the level of an intrinsically disordered protein. So what is an intrinsically disordered protein? It's essentially a protein that has one or more intrinsically disordered regions, meaning segments in a sequence that are inherently unstructured under physiological conditions in isolation. And probably the best way to explain it is with an example. So what you see here rotating on the right-hand side, it's a structure of a chaperone protein. It's actually over a dozen of superimposed structures of the same protein done in NMR. So after you superimpose those structures, you can see some regions where all those structures converge very well. There's this sheet right there in yellow where you can see really good convergence, and the helix at the top where you can see very good convergence. But at the same time, you see those two white regions, one on the right, one at the bottom, where those structures don't converge. You could see this conformational ensemble, a large, very flexible part of the structure of this protein that doesn't really converge to one conformation. And that's what disorder is about. So that's the second part of the definition of the name intrinsic disorder. So I've explained what disorder is. There's the first part of the term, intrinsic. Why do we call it intrinsic? Because the disorder itself is intrinsic to the sequence of the protein, meaning the sequence itself defines the disordered state of a given segment, meaning disordered regions have certain sequence bias. They tend to be enriched in polar residues, hydrophilic residues, charged residues, and depleted in hydrophobic residues. So there's a signal in a sequence that should allow a computational tool to identify disorder in the sequence itself, okay, where those regions are. And why should you care about intrinsic disorder? Well, there's many reasons. I list some of them here. Number one, there's actually a lot of disorder in proteomes, and particularly in eukaryotes. So a number of computational studies done by several different groups conclude that anywhere from 30 to 50% of proteins in eukaryotic organisms have disordered regions, long disordered regions. We call long disordered region long if it's at least 30 residues in length, consecutive residues in length. So it's like a domain size chunk of disorder. Those disordered regions, why they're not structured, they are functional. They implement a host of really key cellular functions, from signaling to regulation to molecular assembly to essentially protein nucleic acid interactions. And this is why they are critical. Those disordered regions are critical for translation and transcription. And recently, over the last few years, people have implicated disorder in the process of phase separation as well. Now, the reason why I'm here is because relatively few of those disordered proteins were characterized experimentally. And what I mean is intentionally characterized disorder, both structurally and functionally. There's only about 2,500 intrinsically disordered proteins that are fully characterized. So there is a need for computational models to fill the gap, to essentially annotate disorder in all the other millions of proteins where the disorder is not yet discovered. So how would a disorder prediction look like? So I'm going to use this example protein. It's CR3P protein. It's a transcriptional suppressor protein. And a part of the function of this protein involves protein-protein and protein-DNA interactions. It's involved in regulation of the structure of chromatin. So it definitely binds DNA. Now, I picked the experimental annotation for this protein from a DisProt database. This is, in a way, an equivalent of PDB for people who do disorder predictions or disorder analysis. So this is a long protein, about 1,000 residues long, and right in the middle, smack in the middle, there's a long disordered region that is key to the function of this protein. This disordered region implements both protein-protein and protein-DNA interactions. And if you would run disorder prediction of this protein, you will get a profile that looks more or less like this. At the top, you see the black line, which are amino acid level propensities. So for every amino acid in a sequence, you'll get a score that quantifies how likely or the likelihood of that residue to be disordered. And as those propensities are typically processed through a cutoff or a threshold, which I denote with this dashed line. So residues with propensities above that score are considered to be disordered in a binary state, disordered versus structured. And of course, that threshold can be adjusted to your desired rate of disorder. So in this particular setup, you can see that at the very bottom, there's a green line called binary disorder that identifies disordered region in this protein. And it's in a pretty good agreement with where the experimental data shows the disorder is in this sequence. That prediction was produced by one of our own tools, FLDPNN. And you may question, well, why did I pick this protein in this predictor? Did you make up that to show how great predictors are? And I would say yes, partly, but I'll try to convince you that in more genetic sense, disorder is predictable from the sequence, and it's inherent to the fact that it's intrinsic to the sequence. So a prediction like that is not uncommon. You can predict disorder fairly well. Now, last year, we set out on a quest to try to document all the disorder predictors that were published throughout the years. And that's a summary of what we found. So this bar chart shows you in blue and green bars, number of methods that were published in a given year. And then the red line is a cumulative number of all the methods. And then on the top of the sum bars, I've mentioned some of the more popular, more known methods. The methods on yellow background are the methods that we contributed. So we contributed measly 4 methods out of a whopping number of 103 in total. So it's a field that has about 40 years of history. The very first method was 1979, with over 100 methods to its credit. Now, if you look at this plot, you will probably find that on the left-hand side, there's very few methods up to about 2002, when there's a sudden spike in the development efforts. And this neatly coincides with inclusion of disorder prediction into CASP. So as of CASP 5, the disorder prediction was added as one of the assessment categories in CASP. And that really motivated people to pay attention to the disorder prediction. And you can see what has happened. About 10 methods, 9 methods, 8 methods in the following years were developed. And the assessment of disorder continued in CASP until CASP 10, and it was discontinued after that. But the field itself has survived rejection by CASP. And you can see that we still developed some methods after that, and eventually in late 1910s, we've developed our own experiment called CADE. So CADE stands for Critical Assessment of Intrinsic Disorder Prediction. So it's an equivalent to CASP, but done by disorder community. And the results of the first experiment were published just last year. So this is what I denote there in blue at the top. And the methods correspondingly highlighted in blue are those that did particularly well in that CADE assessment. So you can see that they were developed quite late in that, throughout the timeline, in mostly late 2010s and 2020s. Now, let me briefly summarize what CADE have discovered. So first, let's set up the experiment. It's a little bit different than CASP in 2 aspects. Number 1, authors of the methods had to deposit their software with the organizers. And the organizers would actually run tools locally on their hardware, which is, in a way, a great punishment for the organizers, but good news for everybody else, because then that software is locked, authors cannot tweak it anymore during the competition, and it's under control of an independent party. So I would say the competition is fair and blind, because authors have no influence over what the methods will produce. The other big difference was the scale. The CADE 1 was done on about 650 proteins, which is about 5 times more than a regular CASP competition. So the result had enough statistical power. Now, you could be surprised, there's only 32 methods that participated, but I think part of the reason why only a third of the field will participate is because authors were required to deposit software, and it's not easy to produce something that works. So about a third of the methods are actually workable. You can install and run them. Now, the other interesting part about that experiment was that besides evaluating quality of disorder predictions, they will also evaluate quality of predictions of disordered binding regions. And I'll comment on both. This slide only specifically talks about disorder prediction quality. So on the left-hand side, you see ROC curves for different top 10 methods. And in a figure legend, you cannot see the cursor. Inside the legend, you see an annotation of A and C. A stands for AUC. So it's like a score that will tell you an overall quality of prediction. And then C stands for coverage, the fraction of the proteins in the experiment that a given method was successfully able to complete predictions. So for most of the methods, they can predict pretty much anywhere, any protein. Some had problems with anywhere from a couple of % to 6% of proteins. So sometimes these methods fail to produce a prediction. And on the right-hand side, you see another interesting experiment, the runtime analysis, which was possible because all the methods will be run on the same hardware by the same people. So you could directly now compare runtime. And you can see drastic differences there. The scale, the time scale is logarithmic. So every marker is at 1-fold or 10 times difference. So you can see some methods are as fast as completing a prediction in a matter of a second or 2 or 5 seconds. And some take hundreds of seconds, meaning hours. The difference is by 3 or 4 orders of magnitude between different tools. Now, there's an inherent conflict of interest with me explaining these results because we participated. We were not the organizers. So I'm not going to comment, except I've used a quote from a commentary article that was published together with the assessment, where these authors, not us, commented on overall quality of methods across the whole competition, including, the predictive performance, the runtime, the coverage, and assessment that was done both based on residue level predictions and also protein level predictions. And they conclude there's 4 key methods that seem to win the competition. And among them, there's one that is at least 10 times faster than the rest. And all 4 of these methods are readily available. They're actually available both as servers and standalone implementations. So if you ever want to have or want to do a disorder prediction, there's a host of tools that are accurate, independently assessed, and available for you. And of course, I will advocate use the very first one on the list. It's the best. And I'm not saying it. I'm just quoting somebody else saying that. Now, I'm going to address the elephant in the room, which is Alphafault 2. A big disruptor for the structure prediction field. Last year, authors of Alphafault made predictions of structure across entire human proteome. And they found an interesting thing, that one of the outputs produced by Alphafault, called PLDDT, which measures or estimates by the network, by the neural network, how well the prediction approximates or estimates the experimental structure. So it's like a quality metric that is at the residue level. And what they found after applying Alphafault to human proteome, the bar on the far right side, is that that parameter has varied widely, from a high score that denotes high agreement to fairly low scores that denote low agreements. And then they reevaluated that for the structural data available in PDB. So in parts of the sequences that are solved structurally, that are resolved, meaning for which the structure is produced, you can see a lot of blue area, high scores, high degree of agreement. For the parts of sequences that were not resolved structurally in PDB files, you see the opposite. A lot of orange and yellow that suggests that the agreement is low. And the conclusion was, well, this score could, in some way, approximate the location of those unresolved areas, which is location of disorder in a sequence. So what happened next? Well, somebody picked up on it and evaluated Alphafault in a CADE data set. And not surprisingly, Alphafault does relatively well. So this is the ROC curve on the left for the Alphafault itself. And then the other panel shows the Alphafault 2 compared to the top 10 methods from CADE. And the Alphafault there is the red line and the red bar right here. So it does relatively well. The AUC score is reasonable, but not as good as the leading method. So I would conclude that, yes, Alphafault is a disruptor, but not necessarily for disorder prediction. The disorder predictor, the meaningful disorder predictor, is designed for that problem, still can outgun the Alphafault in the disorder prediction itself. All right, so I want to switch to like a second part of my talk and now move away from pure disorder prediction and move to prediction of disorder function. So a subset of disordered regions that implement particular functions of disorder. And there was a lot of effort done in recent 3-4 years to identify disordered regions that bind specific partners. So disordered regions that participate either in protein-protein interactions or protein-RNA interactions or protein-DNA interactions or protein-lipid interactions. So like a subset of disordered regions that implement a particular function. And there's about 20 plus of these methods right now available. And I want to draw the attention to this little service that we've developed and published 2 years ago that gives you kind of a comprehensive disorder prediction profile. You enter one sequence, and that piece of web server will not only identify where disorder is, which is the gray line at the top, but it will also grade particular binding partners for those disordered regions. So for this example protein that I've introduced before, if you would run it through the server, it will again identify disorder where it really is, and then say, well, with the green line, it's binding a protein, and with the purple line, it's binding DNA, and with the light blue line, it will say it doesn't bind RNA, and then at the very bottom, there is a purple line. Dark blue binds DNA, and the purple line at the very bottom says multifunctional. And this is a cool feature of disordered regions. They can be multifunctional in a sense that one region can implement a very diverse set of functions. So in this case, it can interact with a diverse set of different molecules. And the way it's doing it is that the disordered region will actually fold upon interaction. And it can fold into different conformations depending on the partner that it binds to. So one region can implement multiple functions. And that's the inherent difficulty with prediction of disorder function. So it's not one function, it's multiple functions. And the structural signal could be confusing because it could lead to multiple different conformational states depending on the interacting partner. So as I've mentioned before, Kate has evaluated prediction of binding disordered regions. The evaluation, it's the very first time the evaluation was done. So in this case, it was done in a partner agnostic way. In a sense saying, this disordered region will bind with some partner, any partner, or it would not. So this assessment has to be taken with a grain of salt because the methods that participated in this assessment, there was 11 of them, they actually work and predict interactions with a specific partner. So they were not suited for that type of a prediction. Still, 2 of them actually outperform a baseline, which means they produce meaningfully good results. One of them is called Anchor, and the other one is called DZRDP bind. Both of them actually focus on prediction of protein binding regions. But at the same time, the other part of the news is not as great because the quality of predictions of binding disordered regions is not as good as the prediction quality of disorder itself. As you can see in this plot, the AUCs drop to the level of mid-0.7s. So the quality is definitely not as good. The runtime, though, is still very good. You can get those predictions in a matter of seconds. So they can be processed on a large scale. And the conclusion from the competition was, well, prediction of disordered binding regions is hard and more work is needed. And following up on that, we've looked at a different at these results from a different angle. Since the overall quality was evaluated, we've asked a different question. Would prediction of disordered binding, meaning binding in disordered regions, be different or similar of prediction of binding that is trained from structured data? Because there's many models that do that from structured data. So we collected a benchmark set, a large benchmark set that essentially is balanced, where half of the interactions are annotated using disorder in disordered regions, and half of the interactions are annotated from structured complexes. And we run those methods through this type of a data set. And this is summarized on this slide. And the very leftmost panel, it's an assessment of 2 types of methods, those in green, which will build or train from structured annotated interactions, and those in orange that were built or trained from disorder annotated interactions. And there's only 2 orange because only 2 of them worked relatively well in Kate. So we picked the best 2. And then there's a black bar that I will come back to later. So if you look at the structure annotated proteins, what you see is structured trained methods do fairly well. And the orange methods don't. But the middle panel that shows the half of the data set that was that includes disordered annotations in disordered regions, the situation reverses. Most of the green methods perform fairly poorly. The orange methods perform fairly well. So if you look on the composite, in the totality, the rightmost panel on all the proteins, you can see that all the green and orange methods reach a limit. And that brings me to the black bar. The black bar is a simple consensus of the best disorder and the best structure trained method. It's a consensus that implements a union of the two predictions. So we take a composite of the two. And you can see, well, there's a visible increase, which would suggest that these two communities have developed methods that complement each other. So if you want to find where protein binding residues are, well, you should use one of the models that is trained from structured data, but you should also resort to using one of the models that is predicting interactions in disordered regions, and these two combined are going to give you a fuller picture. That was 2 years ago. This was done for protein-protein interactions. Last year, we've completed another project where we look at the same thing for protein-RNA interactions, and we see a similar story. The structure-trained methods do well for structure-annotated proteins. The disorder-trained method do well for disorder-annotated proteins. And if you take a consensus, you'll get an improvement. Should the same happen for the prediction of protein-DNA interactions? Yes, it will. I just don't have the corresponding slide ready yet. I have the numbers. I didn't have the time to put it into a figure. Now, with that, I want to conclude this talk by giving you a spill on a couple of resources that are readily available to people who want to use predictions of disorder or disorder functions. Those are databases of pre-computed predictions. So now if you want to use prediction of disorder or disorder function, you don't have to download a method, run it, go to a server, create a profile, make predictions. You can actually go to a database and download already available prediction. And I will highlight two of those databases. The MobiDB, which is by far the largest database that focuses primarily on disorder prediction. This database actually includes results for about 10 disorder predictors and a consensus of all of those, and argues that the predictions that it provides are fairly accurate. And I would agree with that. That consensus works quite well. And it's in a scale of pretty much the whole UniProt, a few versions back. So like maybe 2020, not 2022. Now, I will also discuss the database at the bottom for two reasons. Number one, we've developed it, so I have to have some shameless self-promotion here, of course. But this database offers a different perspective. It provides residue level predictions, not only for disorder, but for a host of both structural disorder-based annotations. So I'll resort to an example, back to the example. If I would apply MobiDB, which is the disorder prediction heavy database to the same protein, you see a very rich profile. At the very top, the blue and red line identifies where disorder is based on this consensus, based on a bunch of different methods, and it does it fairly well. Again, it's in close agreement with where the experimental annotation is. And below, as you go through the lines, you will recognize there is actually a secondary structure prediction here. And what you will find there is depletion in coils and sheets and increased propensity for coils for the disordered region. You can see those kind of a blue, red, and yellow profiles there. There's an Alphafault prediction included for the proteins where Alphafault was run. And you can see here again, that's the PLDDT score, a low value for where the disorder is. And below that line is the LIP line. And LIP stands for linear interacting peptide. And that's essentially a ligand agnostic classification of binding. So this is the annotation of the fact that this disordered region binds a molecule. But in this case, the molecule type is not disclosed. Now, if you would use the ScribeProt as a source, it will give you a different type of an information. On the left-hand side, you see a variety of different predictions and methods that are included in this particular database. So you're going to get a prediction of solvent accessibility, linker regions, binding regions. You'll get a conservation score, you'll get a secondary structure, you'll get a location of signal peptides, a variety of different structural and functional aspects. The scale is much smaller, it's only 1.3 million proteins, but that's pretty much 80 plus model organisms. So the organisms that are the most popular are all included, of course, including human and mouse. About 50 eukaryotic organisms and about 30 prokaryotic and viral organisms included. The reason why it's small is because it's a lot of predictions. It's about 8 billion predictions for that version of the database. Now, the profile that you get out of this database is very different. At the top of it, and I apologize for the small size, you have like a protein level summary of different characteristics, the amount of disorder, the breakdown of secondary structures. And then below that, you get profiles, residue level profiles. So the light green profile shows you, identifies where disorder is. The profile below it is the solvent accessibility, the pink profile. The profile below it is the secondary structures, color-coded, yellow, green, and red, like it's done in PDB. And then below that are profiles for binding regions. And again, if you run our example proteins through this resource, you will find that, well, the disorder is what it's supposed to be, and it's protein binding region. Now, both of these databases give you great flexibility in how you receive the data. You can do it through the profile like that, where you have a way to visually see the information, but you can also collect raw data, raw predictions at the protein level, at the genome level, and at the database level. So if you're skilled, you can parse that data at large scale, essentially. And I pretty much ran out of time. So I'm going to conclude the talk with a brief summary. Intrinsic disorder prediction field has a long history, as probably you've noticed, about 40 years of history, with 20 years of fairly intense research. Over 100 methods were developed already. And the conclusions from the recent assessment are that, well, disorder prediction can be done accurately. The fun fact about the 4 methods that won that competition is that they all rely on deep learning models. Better yet, we've done a little study that looked at all the deep learning methods versus all the methods that are not deep learning based. And what we found is all the methods that are doing deep learning, no matter how good they are, they're significantly better than the methods that don't. Looking at these 2 populations. So deep learning have really deeply penetrated this field. So we became a victim of deep learning as the structure prediction field became a victim. The difference is there are different types of architectures now being deployed to predict disorder. It's not just Alphafault. There's different architectures that can do equally well. Some convolutional networks, some transformer networks, some feed-forward networks. That the architecture is not decided yet, in a way, by the field. The prediction of disorder binding is not as good, but it's at the levels that are usable. And probably more work is needed here. And I want to emphasize the access to the modern resources where you can get full profiles of disorder and disorder functions in one place, or databases where those predictions are pre-computed. So we as a community kind of strive to provide easy and convenient access